Hi, this is Amy Proal with the Polybio podcast, and my guest today is Dr. Ben Readhead. And he's the Edson Endowed Professor of Dementia Research at Arizona State University's Banner Neurodegenerative Disease Research Center. And his work has partially focused on using biomedical informatics approaches to better understand the role of infectious agents such as herpesviruses in conditions like Alzheimer's disease. He's also studying the collective composition and impact of organisms, the microbiome, in the brain in order to better understand how microbe and virus activity may contribute to neurodegenerative disease. So with that, Ben, thank you for joining.
Thanks so much for having me. Great to be here.
Great. Well, I'll jump in. I first saw your paper where you identified several herpes viruses in the Alzheimer's brain, and you got data sets where you collected data from autopsy brains, and you also showed that some of the the presence of the virus is correlated with some of the gene expression that might have contributed to the Alzheimer's disease process. Now, we could discuss that study a little bit more, but before we do, how did you even get there? What made you interested in studying this topic of infection in Alzheimer's disease in the first place?
That's a great question. Yeah, we kind of fell into it. I'd always been interested in brain disease and neurodegenerative disease in particular, but at the time I was working in a lab at Mount Sinai in Joel Dudley's lab. And this was a lab that looked across many, many different diseases at once. And one of the hats that I was wearing and still wear is doing computational drug repurposing. And so this is where you use computers to try and connect high dimensional disease signatures like gene expression and genetic signatures with the universe of drug signatures that we have. And so the idea is, you can start with a disease that you care about, and then you could kind of use this kind of systematic approach to try and see which drugs might shift those disease networks in a way that could be useful or interesting, so you can kind of prioritize your experimental studies. And so we had some friends and collaborators at Mount Sinai who are dedicated Alzheimerologists, like Sam Gandhi and Mary Sado and Michelle Ehrlich. And they came to us and they said, we're thinking about putting together a clinical trial using a repurposed drug that's already in use for some other indication. Is there something you can tell us, about drugs that shift Alzheimer's networks? And that kind of sent us down a rabbit hole, really. We started looking around at what sorts of molecular data were available on Alzheimer's disease. And it's kind of a long, convoluted story, but we eventually found a data set that had been generated by my now collaborator and mentor here in Arizona, Dr. Eric Ryman. So him and his team had generated this data set 10 or 15 years ago now, which was from postmortem brain tissue samples from patients with and without something that they'd called, they called it non-demented Alzheimer's disease. So these were patients that had a lot of neuropathology, but they didn't yet have any dementia symptoms. So the idea was, maybe these are patients that had they lived longer, they would have gone on to develop some kind of dementia. And so we were interested in studying these and comparing them with age-matched controls. And we kind of set about building up some regulatory networks from these two different kind of paired data sets from the same institution. And it was really interesting, kind of what came out of these networks were, firstly, they looked quite different. When we looked at the sorts of genes that were driving the networks in the non-demented Alzheimer's disease, which we ended up calling preclinical Alzheimer's disease in our paper, just for ease of use, even though you could kind of argue about whether or not they are actually preclinical. But nevertheless, the networks already looked very different. And when we started to get to know some of these genes and some of the pathways that these genes belong to, we saw that many of them were actually hitting on viral biology pathways. We were looking at zinc finger transcription factors like SP1. We were looking at microRNAs like miR-155, which have all sorts of really interesting relationships with double-stranded DNA viruses like herpesviruses. And so from kind of a number of different directions, we started to get sensitized to this idea that maybe some kind of microbial or particularly viral perturbation could be explaining some of what we're seeing. But all of these networks, they've been built on old technology, gene expression, microarrays. So we couldn't, there wasn't actually any sequence data that had been collected. But we're at the point where we're saying to ourselves, like, wouldn't it be really cool if we could look at some samples, that have undergone next generation sequencing, where we're actually like sequencing all of the nucleic acids that are present in the sample, And we could just go and look for anything that might have been captured that's not human. And so it was purely fortune that around that time is when the Accelerating Medicines Partnership in Alzheimer's disease, AMP-AD, was starting to ramp up. And so this is, it's a huge effort. It's led by the NIH. It's got a bunch of academic institutional partners, including Mount Sinai, where I was at the time, as well as industry partners in pharma. And it's got a huge data generation component. And at that stage, they were starting to ramp up the next generation sequencing of postmortem brain tissue samples. And so, really, we had access to this data. We started sifting through it in all sorts of different ways, looking for anything that could have been inadvertently captured. So this profiling was done ostensibly to look at host biology, to look at host genetics and host gene expression and all that kind of stuff. But we started to see that in a decent fraction of samples, we were seeing non-human reads. And as we started to kind of mature our analysis, we started looking at differences between cases and controls across different brain regions. And then more and more data was becoming available, so we had additional cohorts to look at. And what we saw in aggregate was what we thought was an increased abundance of several different human herpes viruses in the Alzheimer's cases compared with controls. And there was 3 in particular that came out of our study: HHV6A, HHV7, and HSV1. Although, one of the broader lessons we took from it was that although these ones looked especially interesting, really there was kind of a whole family of viruses that we saw as emerging as potentially relevant to Alzheimer's disease. And I should bracket that to say, none of that is really compelling evidence that any of these things are causally contributing to the disease. But it did give us an opportunity to have a much richer molecular context for these observations. So, we were able to do more than just say, there's virus here, but there's not virus here. What we could also do by virtue of having the host genetics, we're able to build out more sophisticated networks to look at what host genes were associating with these viruses and what was being regulated and regulating the virus. And from that, we're able to get kind of a much richer molecular context. And so some of these viruses, we saw them kind of right in the middle of known Alzheimer's disease biological networks. And so, as I say, it's not evidence that any of these things are causal, but it gives us a much more specific idea of experiments that we can do to follow up to try and better understand what they're doing there.
Yes, that is very interesting. So when you were doing this and you were just looking at those first data sets, had you even read, because there are a few teams that have already, for example, found HSV1 or herpes simplex virus 1 in Alzheimer's brains before, I think, or the herpes virus had been connected to Alzheimer's by a few research teams previously. Did you even know of that work, or was this just an accident and almost on your part, and then later you realized that had already been looked at somewhat?
Yeah, it came afterwards. So really just kind of wandered into it a little bit blindly. It was around about the stage where, where we were looking at some of these kind of viral pathways that were being implicated by some of the network drivers. And when we dug into that and then kind of looked at the broader context in Alzheimer's disease, that we realized that, yeah, there'd been a bunch of investigators looking in this area for decades and decades. And even going right back to the time of Oscar Fischer and Alois Alzheimer, who first described sporadic Alzheimer's disease. They were having correspondence that they were noting the similarities between neuritic plaques in Alzheimer's disease and plaques that are observed in tertiary neurosyphilis. And so, yeah, we realized that there was a much broader and longer historical context for what we'd kind of found ourselves in the middle of.
It's really interesting, because I always figured that the progression would be like, oh, we had noted that earlier research and then we decided to look for the viruses. But it's interesting that you just basically started to analyze the data sets and found, that you seem to be finding non-human or viral reads in them. That's when it comes to what I already really like about your work here is you're already one of the issues, I think, and I guess this is a question. a lot of times when I talk to people about infection and Alzheimer's and herpes viruses, for example, they'll say, well, healthy people have herpes viruses too. So why would they matter in Alzheimer's disease? And you've already addressed that a little bit, where you started to connect the presence or absence of the virus to gene expression changes. Do you think that what else would you do, or do you plan to do to better build on showing, for example, that perhaps viruses or other organisms involved in Alzheimer's are acting differently or doing different things in patients with Alzheimer's, I suppose, than in healthy people? Is that how you see it?
Yeah, that's a great question. Yeah, and it gets something that I think is really important and probably generally underappreciated. I think we need a better conceptual framework for how we think about the modes of activity that a virus or any microbe really can be taking, and that the richness of its interaction with host biology, can take many different forms. One very general set of pointers, I think, is in looking at host genetics and looking at the difference that different host genetics can play on microbiome, not necessarily even in the context of Alzheimer's disease, but just to give an example, so ApoE4, the E4 allele, this is by a long shot, this is the largest genetic risk factor for Alzheimer's disease, and it also is associated with an earlier onset of disease. It's been described, it was reported by Ruth Itzhaki and others almost 2 decades ago that having at least one copy of the E4 allele is associated with higher prevalence of HSV1 in the brain. And Ruth and her team they were very early on this whole idea of microbes and viruses in Alzheimer's disease. But what it highlights is that just the complexity of interactions between genetics, and that's just one loci, the most significantly associated AD loci, but there are many of those. And HSV1 is a microbe that we paid a lot of attention to. No doubt there's all sorts of other ones that we pay much less attention to for all sorts of reasons that have nothing to do with biology. So I think, I think there really is, we kind of need to broaden the conceptual framework we use to think about interactions between microbes and host biology. And I think often there's kind of a oversimplification of the thinking that happens, particularly around this question of microbes in Alzheimer's disease, which is it a cause or is it an effect? And, so just to briefly lay it out, you can imagine a world in which some kind of microbial perturbation is capable of leading to the onset of Alzheimer's disease. We don't know if we live in that world. You can also imagine a world in which maybe it's not capable of causing the disease, but maybe it's capable of accelerating the disease if it's already established, maybe acting as an accelerant. It kind of adds fuel to the fire. That seems at least plausible, I think. And then you can also just imagine a world in which it's, these observations are just of opportunistic passengers, that, a brain affected by neurodegeneration is less able to fight off, opportunistic pathogens. And this thing is just kind of running along the slipstream of neurodegeneration that's being driven by other factors. And so I think there's at least those 3 broad categories, but I think probably the actual complexity of the biology I suspect it's going to be actually pretty heterogeneous. This is speculative from here, but I suspect there's going to be subpopulations of people where maybe some kind of microbial insult is actually capable of contributing to the disease. Other cases where certainly capable of driving the progression. And I think there's probably a lot of microbial perturbations that are probably not interacting with the disease in any kind of specific way. And you can imagine these different subpopulations being driven by different genetic factors or different environmental perturbations like immune priming states and things like that. So I think it's probably going to be a kind of a lumpy, heterogeneous map of what microbes mean to the totality of patients with Alzheimer's disease.
I couldn't agree more. I agree that you can get to probably a common endpoint in Alzheimer's. Perhaps that's the, amyloid beta production, the tau tangles, but that you may get to that endpoint from different, in either a genetic predisposition that's extremely strong, or in people who have less of a genetic predisposition, there be maybe more of the microbial and viral involvement. And also, I think with that, and another thing I've already seen is that you're looking, and I think this makes a lot of sense, not necessarily for a single pathogen as a driver of Alzheimer's disease. Possibly, first of all, there's the possibility that different pathogens may, I think, act redundantly or do redundant, for example, modify human gene expression in redundant ways, in ways that could contribute to Alzheimer's. Or there may be even a collective effect in which different organisms essentially act together. In that sense, the word microbiome comes up, interacting organisms to also drive and contribute to this disease process. So on that front of what I will call polymicrobial involvement, is that something you're pursuing?
Absolutely, yeah, I love all of that. And I think that's quite true. And I tend to think not necessarily in terms of any single organism, but classes of perturbations that have kind of some similar effect on host biological networks. So I think exactly as you say, there's probably a whole set of microbes that individually or even in combination in complex nonlinear kind of ways can twist host biology in states that make it more likely to tend towards molecular pathology and neuropathology and Alzheimer's like state for that person. And so we're certainly interested in looking as broadly as we can. We've got a couple of different approaches that we're pursuing heavily at the moment. We've really been in a data generation phase this past couple of years, just kind of had our heads down trying to generate the next level of data using kind of latest techniques to help kind of step around some of the ambiguities in the area. But one of them is going back and generating much richer microbial profiles from brain tissue samples, using kind of much more sensitive methods than the original RNA-seq that we used to inform our study. And not just using a different technology, but also looking at earlier and earlier stages of Alzheimer's disease. So the data that we were looking at from AMP-AD, that was mainly clinical population. So people that had already they'd come to autopsy having already had Alzheimer's disease, perhaps still living at home, but having had Alzheimer's disease for several years. And what we know about Alzheimer's disease is that it's got a very long preclinical history. So patients, we know or suspect that 2 or maybe even 3 decades before a patient comes to the clinic complaining that he or she can't find her car keys, there are already changes going on in that patient's brain far upstream. So what we're interested in doing is looking at earlier endophenotypes of Alzheimer's disease. So like mild cognitive impairment, looking at patients with pathology but no dementia, as well as looking at other neurodegenerative diseases. So profiling all of these populations in as sensitive way as we can, so that we can build up a much richer picture of what exactly is there, and then at what stage we can first observe it. And so we've kind of got this metagenomics piece that we're excited about, and that's, we had a lot of slowdowns throughout COVID. Some of them still ongoing, but it is starting to ramp up now. And we're seeing some really promising early signs from that. And one of the other big data modalities that we're excited about are looking at antimicrobial antibody repertoires. So this is, in our case, we're looking at cerebrospinal fluid from patients with Alzheimer's disease, as well as these earlier endophenotypes that I was talking about. And what's really nice about this technology, which it wouldn't have been possible to do this even a few years ago, is it can give you a very unbiased readout of someone's antibody repertoire. So historically, like a lot of antibody testing, especially when it's done in the clinical context, it's a little bit of a pot shot. You're looking for a single viral or bacterial protein that the immune system has raised a response against. What this newest technology gives us is the ability to map onto, or to derive an antibody signal against any antigen that any microbe is producing. And so we don't need to know what's important before we go looking for it, which is massively important for a problem like this. And one of the other things that's exciting about this data is it gives us the ability not just to lock into specific proteins, so you could think about looking for signals that occur at the level of, the individual organism, like HSV1. But what seems to be more interesting from our analysis is actually looking for differential signals between specific proteins generated by HSV1. As an example, there's a bunch of viruses that are coming out in this analysis, but lets you lock into individual protein level differences between cases and controls. And what that gives you is then the ability to look at everything that you know about that particular protein and come up with ideas and get clues and inform your experimental design to say, like, what is the biological context of what that virus is doing in a clinical population that would make that protein where it is that we see the signal. And you can even zoom in closer than that and say, not just which viral protein is showing a signal, but which amino acid or which string of amino acids within that protein is the signal coming from. And so it's largely an unexplored frontier, but we're really excited by it. And the nice thing about this data is it can be integrated with the metagenomics and the genetics that we have on many of these same people. So we can kind of build these complex networks that can stitch together these different omics layers in kind of a cohesive whole and build up what I think will be a really comprehensive view of interactions between the microbiome and the brain and host biology in Alzheimer's disease.
Wow, that is very cool. And to clarify, you're getting the data sets from the autopsy brains for the metagenomics, and then you'll get the cerebrospinal fluid from living patients?
So it's kind of a mix. So we've
Mm-hmm.
most of our microbial data is actually from cerebrospinal fluid that's collected at the time of autopsy as well.
Got it. That's
Yeah,
interesting. So it would be basically the same. In other words, and this is a trend that I've noticed, I think, in the literature, is that I don't know if you agree with me. It seems like the pathogens themselves or the organisms tend to persist in tissue, perhaps, so they would be more located if you're going to look for them in the brain tissue. That's why you get the autopsy brain. And they may not enter fluid. They may not enter the cerebrospinal fluid as much, but the immune response that may be able to signal their presence, that the antibodies may be more likely to get in cerebrospinal fluid. So you're going to take advantage of that knowledge to look for the organism content in the brain tissue and correlate it with the immune response in cerebrospinal fluid?
Exactly right. Yeah. So this is a big unanswered question, the extent to which the brain looks like the immune repertoire. We don't really know.
Right.
Because you could hypothesize that having a really robust anti-herpes antibody response, maybe that means there's no herpes in the body. Should we be interpreting that as a sign that there's ongoing reactivations of infection and that it's kind of barely keeping a lid on things? Or is it actually a proxy for a really competent and kind of comprehensive immune response against this virus? So we don't know to what extent we should be considering these things as a proxy for infection or maybe as a protective feature. So we're interested in looking at all of that, and I suspect it'll probably vary on an organism by organism basis, like what it actually indicates. And then the other thing that's exciting about it, I think, is the extent to which these antibody readouts can maybe act as kind of a historical log file for all the past immune exposures that an individual has had, to the extent that they persist in the adaptive immune response. I think that could be really powerful because, one of the things that I often think about with these kind of metagenomics of brain tissue studies is to what extent the most relevant signal is no longer available to be seen. So there's a story I often think about. World War II, there was this statistical research group that was they were doing an analysis of bullet holes, the distribution of bullet holes on planes that were, returning from battle. And they were looking at what they saw is there was a non-uniform distribution of bullet holes across these airplanes. There was a lot of bullet holes on the fuselage, but there was very little on the engines. And so the military was trying to work out, where should we put the armor on these planes? Should we like reinforce the fuselage because that's where the bullet holes are going? And the statisticians looked at it and they said, no, actually you should put them. Basically, the bullet holes you need to worry about are the ones that you never see because those are the planes that never make it back. And so you should be reinforcing the engines. And so, I think there's a version of that problem that I think it's a common problem in biology, but I think it's particularly sticky in Alzheimer's disease, where you got such a long preclinical phase. And so, if we're trying to interpret the molecular pathology of a brain that's been affected by a decade of clinical Alzheimer's disease, it's by that stage, unfortunately, that there's so much compound interest. There's so many things that have gone wrong, and then there's compensatory efforts by the host biology, and then insult upon insult multiplied by compensatory efforts, and it's kind of like trying to untangle a car accident 200 yards down the road, when what you really need to be paying attention to is like why the brakes failed, further up the street, and so I do wonder that these sorts of brain metagenomic studies, one of the questions that we're interested in looking at is like, what's missing, like what should be there that we just don't see. And it might be a red herring. It might be that we can still pick up traces of the most relevant perturbation, but that is a class of vulnerability to this kind of scientific problem that I definitely stay mindful of. And antibodies, I think, are a neat way to perhaps fill in some of that blind spot a little bit.
Absolutely. And I think you're thinking about it in the right context, because you understand that there's a cause and effect, but that there's bidirectional relationships between organisms and the immune system and organism. In other words, it's a constant process. Whenever there's an organism activity, the immune system responds, but then the immune system may, respond in a way that modulates how the organism survives. And so you have to understand that dynamic relationship, because there are some teams that sort of assume that the immune system is always the enemy. In a sense, when they see something that's immune related, they go, oh, that's a negative, right? Look, there's more antibody, there's more cytokines, there's more X number of cells. But you have to understand, as you do, that those cells have a function as well, that antibodies are generated as part of immune defense. So teasing out, exactly how the immune system and the organisms are connected, and not assuming that it always goes just one way or just the other way, is really part of understanding the picture. That is a really good way to approach it.
I think it's one of these common failure modes in biology to assume any one thing is always good or always bad.
Yes.
I think probably you're familiar with this, but Rudy Tanzi and Robert Moyer's theory, the antimicrobial protection hypothesis. This is a really nice example of, so for anyone that's not familiar, it's this idea that molecules like, amyloid beta are an innate immune effector molecule that's capable of essentially enmeshing and entombing invading microbes, and then they lead to the release of reactive oxygen species and essentially neutralize invading pathogens. And that there's some part of Alzheimer's pathology and eventual dementia that is essentially the collateral damage caused by that compounding over many decades in a brain tissue system that has really limited capacity for repair. Yes. And so the prediction there would be that you probably don't want to turn amyloid generation down to zero, because it's this massively evolutionarily conserved molecule connected to this massively conserved immune pathway, and so it's not doing nothing. And so, I mean, it's, to paraphrase what I've heard Rudy say, that you wouldn't want to turn it down to zero. You'd want to dial it down a little bit, and much more in kind of a preventative context than in a treatment context.
Yes. No, I mean, I was almost, my next question is, I knew Rob Moyer. He was actually on one of my grants. Unfortunately, as you know, he passed away, but now 2 years ago or more. For those listening, Rob Moyer was a researcher. He was at Harvard Medical School working with Rudy Tanzi, who's still working on this topic now, again, also looking at infectious contributions to Alzheimer's disease. But what Ben mentioned is they were looking at the amyloid beta, what many people call the plaque in Alzheimer's disease, the amyloid plaque. And they were looking at it, especially Rob was saying, maybe this plaque has a function. Maybe it's not just this waste product that keeps accumulating that just has no purpose. Maybe it's doing something. And that was based on the understanding that we have, zebrafish make amyloid. In other words, this is a very highly conserved, part of the the molecule or whatever. So he thought, like maybe as a function, because if something is that conserved, it usually will be weeded out by evolution from becoming in part of more, species if it doesn't do something. Yeah. And so in other words, he showed, and it's more than a hypothesis to me, he's actually, that lab has shown that this plaque in Alzheimer's brain may actually be an antimicrobial peptide, part of the innate immune system that forms in response to pathogens or other basically environmental insults. And so that really, sort of turns Alzheimer's, a little flips the way we look at Alzheimer's, but it also makes a lot of sense because, like you said, there have been, or as you alluded to, we've been doing drug trials where we've been removing amyloid from the Alzheimer's brain. I think there's hundreds, if not thousands of these trials of just amyloid removing drugs. But if amyloid has a function, especially an immune function, then as you were mentioning, we may not want to remove it right away. We may want to think more strategically about how and when we would remove amyloid. In other words, if it has a role in protecting against organism, we might want to let it do that for a while and remove it. I don't know. You tell me, I guess. Yeah.
Oh, I wish I knew.
I know.
But yeah, I agree with the trend of what you're saying. And I mean, just about Rob, he's sorely missed. I wish we had 10 more Robs in the world.
I know.
He is a fantastic scientist and a wonderful person. And also grew up in Western Australia, so it's a point of pride every time I hear his name come up. But yeah, I think it's, this idea of Chesterton's fence, which is the idea that something that's in place, you should know why something's there before you remove it. You should know what's the motivation for it to be there. I know that evolution has a motivation, but it has evolved to complex. These molecules have evolved under complex selective pressures, and as best I can tell, I mean, Rudy's reasoning on this makes sense to me. That you wouldn't want to hit it through the floor, but probably dialing it down a little bit is probably fine. But I mean, time will tell. I mean, I should say I'm not an expert in A beta physiology or Alzheimer's trials, but that to me makes some sense. That I think a lot of the dynamics of Alzheimer's disease are kind of happening at the margins. That a reduction of of some of these critical pathways by 10%, say, that could extend out the time to diagnosis by even just 5 years, which simulation studies have shown that even that would be just massively beneficial to countries like the US in terms of essentially would shift a huge amount of the Alzheimer's disease burden later in life, when by which time other diseases then become the priority to find treatments for. And so I think it may not have to be a dramatic intervention, especially if it's done in a preventative context, to be a difference that makes a difference in terms of how this kind of late stage disease plays out.
Yes. Okay. And, in addition, so your one paper focused on herpes virus activity in the Alzheimer's brains thus far, but then other organisms have also been implicated in Alzheimer's development, bacterial pathogens, fungal as well. Have you found any of those organisms? I don't know if you can talk about it yet, or in any of the brains you're looking in thus far. Do you think we're looking at viral contributions, bacterial contributions, fungal contributions, in other words, different types of organisms being able to contribute to the disease?
I think it's certainly possible. I'm really interested in these other kingdoms as well. But so we do have, so many of my projects is the way they've evolved. They tend to be fairly virus centric. Having said that, a lot of our metagenomics that we're doing at the moment, we're intentionally looking broadly, just doing shotgun metagenomics in a decent fraction of the samples that we're looking at. And so with that comes the ability to pick up, all comers, anything that has nucleic acid. Some organisms are easier, some are more difficult to pick up. And we certainly are seeing signs of other like bacterial organisms, particularly intracellular bacteria, things like that. Whether or not they're case associated yet, I don't know. We're still kind of in the data generation phase. But yeah, it certainly seems possible that that there's been a lot of good research done by other investigators looking at bacterial pathogens, fungal pathogens, and to me that all sounds plausible. And I think what's also becoming increasingly obvious is the importance of the gut-brain axis as well. So the potential for microbial activity to be affecting central nervous system function, but in a way that might not necessarily be dependent on the microbe actually making it to the brain, but having these other intermediate metabolites, like bile acid receptors and things like that could be making their way to the brain and twisting biology in that way.
Right.
So I wouldn't rule anything out.
Agreed. So even organism, in other body sites, whether it's via vagus nerve signaling from the gut to the brain or from other body sites, could potentially, lead to activity in the brain, even if the organism isn't there. I don't see why not, right? I'm sure it's a combination of all of that activity at the end of the day.
And the emerging P. gingivalis story is
right.
Is another example of this, where potentially we've got these gingipanes, which are these molecules produced by the organism, but they're not the organism itself, making its way into the CNS and potentially causing AD-like effects in the brain.
Right. So that would be P. gingivalis, which is a common oral pathogen, right?
Exactly.
And there's a team that found that. Did they find the P. gingivalis in the brain itself, or just the gingipanes? The gingipanes are the proteins created by P. gingivalis.
Yeah. So as far as I know, they didn't see it in the brain. I think it was more. They were more positing that it was being mediated by the gingipanes.
Interesting. In other words, the organism doesn't necessarily have to be in the brain. It's a product or something that it creates may get into the brain, and that may also be a driver of disease.
That's right. Yeah. And there's a similar class of inquiry that I'm seeing picked up by other investigators as well, which is looking at novel mechanisms whereby peripheral infections could be impacting on CNS function. So herpesviruses, so HSV1 infections as an example. Most commonly these are infections that are laying dormant in the trigeminal nerve, which is reactivation happens there, and it seems reasonably clear that at least some of it makes it into the CNS, but there are investigators that are trying to carve out novel mechanisms that simply require that the reactivation happens in the peripheral nervous system, but then in a way that can impact on the CNS.
Interesting. And that would just be through nervous system communication potentially?
I'm not really familiar with the deep biology on that one. I'd have to get back to you on that. But it is an interesting general class of investigation, like what mechanisms could account for a kind of a peripheral to central interaction like that.
Yeah. That's have you heard of the illness Myalgicencephalomyelitis chronic fatigue syndrome, or ME/CFS?
Sure, yeah.
Okay. So that's the illness that I study primarily, along with several colleagues, and it has so many overlaps with Alzheimer's and the work that you're doing. And these are younger people, often, doesn't matter, but people who can be, even children. And the average case, or many cases of ME/CFS, start with a viral infection, including herpes virus infections. And then and then people go on to develop chronic symptoms that do not go away, that become very debilitating. And, they actually have a lot of cognitive dysfunction. So there is some of the symptoms. So you go, well, interesting. I mean, I feel that are certain really overlapping trends, which is that we, in this condition, we do think that the pathogens that initiate cases may, at least in the case of the herpes virus, which is by definition a persistent pathogen, but there are also some of the other viruses may not fully clear, but if they stick around and they're drivers in some of the symptoms, they may get into the central nervous system. Or they may be in the periphery of the body, but vagus nerve, which innervates a lot of different body sites, may be able to sense inflammation associated with their presence and sort of convey that to the brain stem, which is, part of the brain that controls a lot of sickness behavior, response, and autonomic function. In other words, you may have, like you say, it's possible for an infection to not be in the brain itself, but to signal at least via vagus nerve, which is something we look at to be able to alter the way that the central nervous system is itself responding. So it's a really similar trend. And I think that's part of why I started to follow the work on infection and Alzheimer's, because we face similar challenges. One of the issues that we have with ME/CFS is that which thank goodness, people don't actually pass away often. So it's harder to get autopsy brains. So we cannot do as much, and we're trying to set a better postmortem collection up over time. Now, if you, and I don't know, this might be a tough question, if you had to study the impact of infection on the Alzheimer's brain, and you could only do it in living patients, how would you try to go about doing that?
That's a great question. Yeah, because it really forces you to think creatively about how to connect, these microbial traits with the clinical traits that we care about. So there's so I think antibodies, like antibody repertoire profiling and T-cell repertoire profiling, would be really interesting things to pursue. I think also, one of the things that I think is really missing in Alzheimer's research, or rather one of the missing ingredients that I think would make our life a lot easier in resolving some of these questions, are having more elaborately phenotyped microbial, so electronic health records that do a better, more comprehensive job of capturing microbially relevant perturbations. So at the moment, electronic health records, they're really optimized for some combination of clinical record keeping, but also billing as a major driver of what it is they're incentivized to capture. And so if I think of the Alzheimer's example, many of the epidemiological studies that have emerged showing associations between different herpes virus infections and eventual progression to dementia, which there are several studies like this. Really, I think it's a version of the looking for your keys under a lamppost, because that's the only place where there's light. And so if I had in a perfect world, we'd have a much more comprehensively phenotyped set of infectious exposures that we could then associate with all the clinical and neuropathological phenotypes that we care about. So that's, I guess that's the resource that I think we should be trying to build some proxy for. And so antibodies, antibody profiles and T-cell repertoires and things like that, and maybe immune cell profiling and cytoff, and high dimensional readouts of immune system activation states. I think all of these things are potentially can give you a potential set of tools and approaches that could let you get at that in the absence of being able to sample brain tissue. And then there's maybe I'll leave that.
No, that's part of what we're doing, trying to infer what's happening from the immune system side of it, right? And then to the extent that we can, sometimes collecting tissue from surgery or from biopsies that may contain tissue that you cannot get from a living patient, and then also trying to get more brain collection to happen over time. The antibody profiling, the unbiased, and by unbiased, I'll explain to the people listening. A biased approach is when you have to know what you're looking for to find something. So if you want to find an antibody to a certain pathogen, you have to say, I want to find this herpes virus, and then you find it. But what Ben is developing for antibody detection in cerebrospinal fluid, it can identify any antibodies in that sample without you having to decide ahead of time what you're looking for. It'll just say, this is what's here. So that's a huge ability to be able to explore more and say, oh my gosh, maybe organisms that we don't even know are involved in this. We can actually find that out by using these approaches that we don't have to know what we're looking for ahead of time. That's a really cool thing you've created. I actually can we, if it works out, can we send you some ME/CFS cerebrospinal fluid one day?
Yeah, I think that'd be a great idea. So look, I just to be clear, I can't take it. I can't take
I know.
credit for having developed the technology. It's a startup that we've been working with, kind of a collaborator called Seramune, who developed this serum epitope repertoire analysis.
Nice.
But yeah, I'd certainly be interested in brainstorming on ways that we could use that kind of technology for ME/CFS. Yeah.
That's great. Yeah, it's really interesting. And I guess then going back to, you recently gave a talk to the NIH on brain microbiome, but I wasn't able to watch because I had a conflict. So when you talk about brain microbiome, how do you define that?
Well, fairly simply. So, I mean, the way in which I use it, and others may have different definitions. Really, it's just any set of organisms, the collective set of organisms living on or in the brain that are capable of interacting with host biology. And so, there may be some sense in which some of these are even kind of commensal organisms. Often I'm thinking about it in terms of potential pathogenic relationships, that is, say, the relevance to Alzheimer's disease.
Right.
But yeah, that's kind of my working definition.
That's mine too. It's just organism where there's more than one, and they usually are interacting with the human genome, likely. Because I think there's been some pushback on the possibility that there's a brain microbiome. Obviously it's somewhat of a paradigm shift because we've historically thought the brain is sterile. We've thought a lot of the human body is sterile. That now that we have better technology, as we're really, especially the computer-based tools that you use, we're realizing that most of the human body really doesn't seem to be very sterile. But in the brain, that's a real shift. So people go, oh, and I think also with microbiome, sometimes the definition of microbiome that people use is they think of microbiome as beneficial organisms. And I've tried to push back against that a little bit, in the sense that I think that is where some of the confusion on brain microbiome comes from. People say, well, what are they doing in the brain to help us? And it's like, look, they're just there. They're just acting. Microbes don't think every day, how do we help the host? That is not the way I necessarily look at microbiome.
Yeah, it sounds like a fairly human conceit to assume that that these things are required to help us. I mean, I could believe that some of them are adaptive so the studies showing that chronic colonization of mice with, I think it's cytomegalovirus, tunes the immune system in a way that's more protective against certain sorts of bacterial infections. And so I could believe there's some kind of adaptive relationship there, but I haven't seen evidence for it, and I don't think it's required. So yeah, I have sensed a little bit of that, filter being applied to microbiome. But if we're not allowed, if that's called that, then what do we call just the full set, absent of any judgment as to whether or not it's good or bad for us? We need a name for that.
I think it derives from the gut microbiome, where people are most familiar with that vast organism, communities in the gut. And there, yes, organisms do perform functions. They create metabolites that sometimes become part of, we use the metabolite, our human bodies do. Even there, though, there's no intent on the part of microbes to be doing this in some sort of beneficial way. It just so happens they create a metabolite, and that has evolved to be part of, how we use our signal. So I think, I do think it will be helpful for the definition of microbiome to move beyond just symbionts, in a sense, towards just organisms and what they're doing. I don't know. That's one of the things that I regularly end up sort of pushing back on and explaining to people. So in the microbiome, are you looking at healthy brains as well? Do you think that healthy people also have organism in their brains?
Yeah, I think it's great question. So yes, we are looking at healthy individuals. That is to say, aged controls that didn't die from Alzheimer's disease and didn't die of any neurological disease. And we're looking at neuropathology, or some amount of Alzheimer's-like neuropathology is actually really common in the aged brain, even if you don't have dementia. And so we have sets of controls that have some amount of neuropathology, but no dementia. And we're thinking about them as distinct from what's actually a smaller subset of people that have absolutely no neuropathology and also don't have dementia. And so we're kind of mindful of these two, and I might not be right to be talking about it as two classes. Really, it's probably more of a continuum. But we're thinking about this as kind of an axis along which we need to be comparing the clinical populations to. And yeah, I think so. From our own data, yeah, I think certainly we've got spot instances of microbes, particularly viruses that we're seeing. Like a lot of adenoviruses, herpesviruses, in cases and controls. And we're kind of holding off doing the comparative analysis between the groups for now, but until we've got kind of more data generated. But we certainly are seeing the presence of microbes in the brain tissue. Having said that, what we're looking at in this current instance is whole brain tissue. So that also includes kind of a glymphatic space. It includes the tissue parenchyma itself, the cells and the neurons and so on, and then also the vascular space. So it is possible that some of the signal that we're seeing is actually being seeded there from within the blood vessels that are captured within a kind of given tissue sample. I don't think that's likely to explain kind of the whole signal, but I just say that as a bit of a qualification.
Yeah, that seems reasonable. But even if you were to find, for example, a certain virus in a non-dementia brain and a certain virus in a dementia brain, it doesn't necessarily mean that they're acting in the same way, right? That's what you'll further pursue, is that perhaps in the brain with the person with Alzheimer's dementia, they're now modulating human gene expression differently or something like that, right?
Exactly right. Yeah. So this is why I'm in love with kind of multidomic integrative analysis. So that's where my particular bias is, collecting as many different data layers as we can on a set of individuals so that we can look at levels of like microbial readouts, like metagenomics, but at the same time have gene expression data, like host gene expression, proteomics and metabolomics and all these other layers. So we can get a much clearer idea of, we can go beyond that kind of one-dimensional, is it there or is it not there? And say like, who is it talking with? What's it doing? What's its network neighborhood? And get a much richer idea of, what its operating context is.
Yes, exactly. Is sometimes, I'll describe an organism that can contribute to a chronic disease process. It's a little like a person. It can be, behaving, or it could act out. People can sort of change the way that they act. Well, so can pathogens, so can organisms. Even, organism in microbiome communities, they're pathobinds. They can change their gene expression from a commensal organism. They can evolve to be more pathogenic. So it's really the looking at the activity of the organism, the gene expression, or the way it's impacting human gene expression, I think exactly like you're doing, where we're going to find more answers as to what's really going on.
Just like one example of what I think is a really positive trend that I've seen in the field is an increased amount of attention being paid to potential mechanisms that, where a virus could be interacting with host biology, but during a latent state. So, probably most people working in this field would say, it's Alzheimer's disease it's not an active infection. It's not encephalitis. It's not kind of a chronic active fulminant infection. To the extent that any of these microbes are contributing to Alzheimer's disease, it's either a very slow, low copy, indolent infection, or maybe even being mediated during a latent state. So where, one of the remarkable things about herpesviruses as a family of viruses is their ability to use all sorts of sneaky ways to maintain latency within a cell and kind of evade the host immune response. And what I've seen are several investigators looking at really interesting different mechanisms whereby, so to give an example, at the NIH AD microbiome workshop that was held recently that you mentioned, there was an investigator, Dr. Chris Proal from University of Rochester, and him and others in his lab had been investigating the effects of the herpes 6 virus gene U94. So U94 is like the latency transcript for HHV6. So this is the main one that gets transcribed when the virus is integrated into the host chromosome, which is how this particular virus stays latent. And what they were able to show was expression of this U94 gene, and no other viral gene was capable of affecting the ways in which synapses would mature and form, affecting the ways in which neurons were maturing, and the ways in which oligodendrocytes, which are these very important cells that kind of wrap around neurons, the ways in which they were maturing. And it also seemed to accelerate the deposition of amyloid plaques in the brains of these mice. And so it's really different from the model of requiring that a virus comes out of latency, gets encapsulated, and then starts replicating and spreading, from cell to cell. The fact that it seems like these, at least one of these molecules that's steadily transcribed during latency is capable of twisting, host biology into an Alzheimer's-like state. And so, whether or not HHV6 ultimately has anything to do with Alzheimer's disease, time will tell. But I think it's really, a really interesting class of inquiry, trying to connect these latent mechanisms with Alzheimer's disease pathobiology.
I just could not agree more. Exactly, because I think we're being too strict on latency versus active in terms with viruses. And people say if it's latent, it's not doing anything. If it's active, it's going to kill you. Right. And there's these two extremes. And that's part of what I think has led to, there's a pushback on, and the topic of infection and Alzheimer's disease is the paradigm shift occurring now more, although it's certainly been a topic for a long time, but there's still pushback on the idea. And part of that, I think, stems from the fact that people say, well, if there's an organism in your brain, you'd be dead. Right. And what you're saying is, no, look, there can be, subtleties in this. It can be a very low amount of organism. It may even be still in a latent state, but even in a latent state, it can still act in ways that may contribute to disease. And I think that is very important, because it's exactly, we keep going off this, pathogens having to be incredibly active to drive disease. And I know there's a number of herpes virus proteins that are expressed during latency. So I think that's a really encouraging, truly, like you said, to hear that the field is moving in that direction. I'm glad. I'm bummed out I missed those talks, but yeah, they sound good.
It was actually, it was a really fantastic quality workshop. So it was 2 days. Really high quality, really collaborative and collegial, and all sorts of dissenting opinions, but very constructive. And so I think originally the NIH wasn't going to release the video of it, but there's been a lot of popular demand for it, and so I think there will be copies of it made available. So if you want to catch up on it afterwards, I think it will be available for you to take a look at.
I do. All right, I'm glad to hear that. Will Emer, I think you know him, who took over Rob Moyer's work. He's on the scientific advisory board of PolyBio, the organization I run. So he spoke there, and I said, well, where's your talk? I thought because it was NIH, it would be publicly available. Then they weren't for a bit, but it's exciting. I hope that they do make those talks publicly available. But
I think it will. Will gave a fantastic talk. Yeah, he did.
And it was encouraging that the NIH did that, because historically the NIH hasn't been funding too much research on infection and Alzheimer's. How has that changed? That's improving in your mind?
Yeah. And I took it as a really positive sign. Firstly, that the NIH ran the workshop, and at least informally, I get the sense that there'll probably be follow-up workshops. I think it was found to be quite productive. There was an RFA for investigating the infectious etiology of Alzheimer's disease. And they did a nice job organizing the sessions as well. So, at the beginning, one of the early sessions in the workshop was a series of back-to-back talks, essentially from authors that had looked at the same or similar data and come down on different sides of the debate. And so there was a lot of opportunity for back and forth. And, so this included me talking about, our study, our neuron study, and some of the work that we're doing since then, in combination with Dr. Stephen Jacobson from the NIH. So him and his team, following up from our study, they did an investigation in some stored DNA samples from some of the same patients that we profiled, and they did some computational analysis and essentially didn't detect much of any virus in these samples, including, detecting very low rates of HHV6, less than 1%. And so, that was a finding that was discordant with ours. And so there was a lot of opportunity for kind of back and forth between investigators and for everyone to talk about kind of what the shortcomings of their particular approaches are. And I think overall, I found it really productive.
That's great. That's really good to hear. I'm so glad that the NIH has not funded infection, but now it seems like they're changing that. There's an RFA. This is exciting to see that move forward, because I know a lot of research teams have been funded by private foundations or donors thus far. And so it's cool to see the NIH jump in a little bit more.
Yeah, absolutely.
Which is good.
I mean, personally, we've had a lot of great support from the NIH, which I'm very grateful for, but also philanthropy has been a massive part. Groups like the Denta Foundation have been just like indispensable, I think, in catalyzing some research that would be difficult to get funded elsewhere. But I think it's going to, so that they've been really generous in supporting some of our work to look at earlier Alzheimer's related endophenotypes, brain tissue from people with mild cognitive impairment and people with pathology, neuropathology, but no dementia. So we can get a better idea of, to the extent that we see any differences between cases and controls, at what stage are we seeing that emerge?
Yeah.
They've really filled in like a vital gap there.
Cool. Well, that's really encouraging then. Well, then I guess I'll just leave you with, if you were giving advice to other research teams who are trying to jump into this topic, infection and just neurodegenerative disease in general, what would you tell them?
Look, it's a fascinating area. It really is the watershed of a lot of different disciplines at once. And so, I mean, as a general statement, I think we need to be encouraging heterodox approaches, like a lot of different approaches, some more conservative, some more innovative and speculative. We need to be getting the help of career Alzheimerologists. We need to be getting data scientists and bioinformaticians, infectious disease experts, epidemiologists, human geneticists. All of these different disciplines have unique approaches and tools, at their disposal, and any single approach in isolation is limited and has blind spots. But part of the reason why I love an integrative approach is that these blind spots tend to be in different places for different approaches. And so if you can kind of combine them together in different ways, it can kind of fill in the blind spots a little bit. So if you've got an epidemiological study that shows that, and just to give an example, an epidemiological study like the ones that came out of Taiwan a few years ago, showing that a history of severe HSV-1 or HSV-2 infection was associated with an increased risk of progression to dementia in the following 10 years. And then what was really interesting, they showed that all those patients that had the severe infection, if you stratify them according to whether they did or didn't receive antivirals, almost all of that increased burden was carried by the people that hadn't had antivirals. So the flip of that is to say that antivirals seem protective against the risk that herpes virus confers upon like a diagnosis of Alzheimer's disease or dementia. And so that was a fascinating study. One of the criticisms you can raise of it is, maybe antivirals are a proxy for just having better healthcare in general. And so maybe that's a subpopulation of people that are going to do better, if for whatever reason, maybe better socioeconomics or who knows, but do better along a range of health metrics, not just Alzheimer's disease. And it could be a proxy for just better healthcare, which is a reasonable action. And it's something that's difficult to resolve from within an epidemiological study. But if you take that and then you look at those organisms and then you do a molecular study, like maybe you do some infections in brain organoids, and then you're able to show that, they're perturbing molecular networks that are enriched for Alzheimer's disease genes, then you kind of filling in each other's blind spots. Use the epidemiology to tell you what virus or what microbe to pick for your molecular study, and then your molecular study lets you look at connects to the Alzheimer's genetics, and there's ways to kind of dance around between all of these domains to build in a much fuller picture than if we restrict ourselves to just one.
Absolutely. So just break down the silos, use as much data from the real world to inform the studies that are done that you choose to do in the lab.
I think
To
so,
connect
yeah.
people with different skill sets. I think that's what most illness communities need right now at the moment, the integration of that. But it's encouraging to see you describe that in Alzheimer's. So that's great, Ben. It's been really interesting to talk to you. I wish you the best of luck with your research. I'll be watching moving forward.
Thanks so much. I really appreciate the great discussion and the great questions.
Cool. All right. Thank you.
Thank you.